Mountains are currently one of the most threatened ecosystems on Earth. Because most organisms in these ecosystems are only able to be active for such a short portion of the year, rising temperatures and changes in rain or snowfall can have huge impacts on alpine species. Anyone who spent time in the mountains knows that these ecosystems can have an incredible diversity of plant life, including wildflowers. You can find different types of flowers open in different types of places in the wilderness. Avid and frequent hikers will also know that you can find different flowers open at different points in the summer. The timing of the life cycle of organisms, such as when plants will open their flowers, is a branch of science called phenology. You can think of this as the study of the internal clocks of different kinds of organisms. When researchers talk about the threats that mountain ecosystems face, wildflowers are among the organisms they're most concerned about. Because growing seasons are so short and environmental changes are so drastic over the course of the year, wildflowers in mountainous areas are especially tuned to cues from their environment, such as changes in the amount of sunlight they receive. These plants rely on these cues to start or maintain their internal clocks. This is one of the reasons why climate change threatens these plants. Warming temperatures, longer summers, reduced snowpack, all of these factors can change the cues, throwing plants off of their typical schedules, influencing their survival, growth, and reproduction. So, we know that environmental cues are important for plant phenology, but we often forget that mountain landscapes are incredibly diverse. Mountains contain a lot of forest cover, but these forests can vary in the density or types of trees, which influences the amount of sunlight reaching the forest floor. Interspersed throughout the forest are meadows and clearings with abundant sunlight. Other subtle factors, like elevation and slope, can also produce subtle changes in soil moisture and soil temperature. When combined, these factors can produce drastically different environments for different plants, even plants that are just a few feet away from each other. These fine-scale environmental differences may produce different cues for different plants in the same population, potentially disrupting some of their internal clocks and important biological processes. This means that local environments may affect plant life cycles, as well as influence plants' vulnerabilities to climate change. Hi, my name is Scott Nordstrom, and I'm a PhD student at the University of Colorado in Boulder. I've studied the ecology and evolution of populations of wildflowers. Typically, when ecologists think about populations, they think about them existing in a single homogeneous environment. But there's still so much that we don't know about how these small-scale differences in the environment can influence plant biology, and how that influences population vulnerability to climate change. For the last 2 summers, I've been following the timing of flowering of wildflowers at the University of Colorado's Mountain Research Station, located in Boulder County, adjacent to the Indian Peaks Wilderness area. I focus specifically on this area, called the Elk Meadows Complex. Here, I was focusing on 2 species: spreadfruit golden banner on the left, and the slender purple harebell on the right. My main question for the last 2 years has been, how do differences in light, temperature, and moisture influence when these plants open their flowers? And what impact does this have on the ecology and evolution of populations of these plants? Thanks to generous support from the Indian Peaks Wilderness Alliance, in the summer of 2021, I was able to construct and deploy 15 homemade environmental data loggers within these meadows. These sensors took solar radiation, soil moisture, and soil temperature readings, which can be used to create fine-scale environmental models in the study area. Here's a video of one of these sensors in action. I'm here at plot 48 at Elk Meadows, and this is one of my data loggers. In this Tupperware is the main data logging component. This green board is a Mayfly Arduino board that is controlling the probes and running a script that takes measurements. That little black thing is a micro SD card that's storing measurements. And this runs off of 3 AA batteries that last for about 2 weeks at a time. There's a mess and a jumble of cables in here. One of those cables leads to this Apogee SP510 pyranometer, which every 10 seconds is taking a solar radiation measurement as sunlight filters through the trees. Another one of these cables is getting run through this steel pole all the way down into the ground, and it's connected to a Terros 11 soil moisture and soil temperature probe buried about 6 centimeters in the soil. This is taking moisture and temperature readings every 15 minutes. I placed 15 of these sensors throughout the study area. These sensors span a gradient from open meadow interior to partial canopy coverage to full-on forest. Forest types differed among aspen, lodgepole pine, and mixed tree cover. Here are readings from these 3 sensors, all on the same day. The meadow plots, as expected, had full, uninterrupted sun, but under canopy, patches of sunlight are much more sporadic. Here, plants would be lucky to get a full hour of direct sunlight over the course of the day, and often they'll go much of the day without any direct light whatsoever. We're using these sensor readings to estimate solar radiation for the entire study area. This includes using drone imaging to make a 3D point cloud of the forest, as you're seeing from this video here. The drone and sensor images can be combined to provide fine-scale solar radiation measurements like this. This graphic shows estimated solar radiation throughout the meadows on a cloudless June day. Here, you can easily differentiate the open meadow interiors from the dark forest understory. We similarly plan to build fine-scale models of moisture and temperature using data collected this summer. Here's a sneak peek of this data. Here is soil temperature for 11 sensors over the course of one day. All of the plots show a similar pattern, cooling at night and warming during the day. However, you can see that the orange curves for open meadow interiors show more extreme conditions. That is, warmer temperatures during the day and much cooler temperatures approaching freezing at night. The plots that are below forest canopy are much more buffered from the daily environmental conditions. Here are soil moisture readings for the same plots over the course of 6 days. Here, the data will be more complicated. We can already see, though, that for the most part, soil moisture is stable over time, although there's certainly variation from plot to plot. In the spring of 2022, I'll put these sensors out again to capture environmental data at the really crucial time when plants are first emerging from the soil. This will be combined with information about pollination and measurements of seeds, which will help us connect light, moisture, and temperature to plant performance and reproduction. Climate and environmental change threaten plant populations in our own backyards, in part by disrupting their phenology. However, we're showing that not all alpine environments are the same, and that even across short distances, plants can experience very different conditions. Will some plants be less affected by warming temperatures than others? What could these environmental differences mean for reproduction and survival? Many of the charismatic species of Colorado's mountains may be able to persist under climate change better than we think, although it might be that this is just in a few rare pockets of good habitat. Thanks for listening, and thanks again to the Indian Peaks Wilderness Alliance for helping to support my work.